For more information on tutoring, personalized video solutions, or how to support Moof University and the production of more videos, check out moofuniversity.com. Thank you and enjoy. Okay, so metabolism of amino acids occurs, at least for the most part, in the liver. Okay, so how does the protein that we eat become the amino acids metabolized in our liver? That's what this video is basically about. Now, just as a forewarning, there are tons of perhaps unnecessary details ahead. You may need to know them for whatever you're studying. You may not. I don't know. But just brace yourself. Okay. So the first question here is, in which organ does protein digestion begin? And that is the stomach. So we're going to take in food through our oral cavity. It's going to go down our esophagus, right, and into our stomach. And that's where the action gets started. And then we have some digestion occurring in the small intestine. Okay, so this is just an overview of the actual organs involved in the digestive system, but not as important as what we're getting to here. All right, so hope you've braced yourself for the details. Okay, so we're going to go through this step by step. First things first, step A, we start off with this dietary protein here. And it's coming down from the esophagus into the stomach. So here, protein arrives to the stomach and stimulates G cells of the gastric mucosa to secrete gastrin, which is a peptide hormone. Okay, so that's shown basically right here. Just the arrival of the protein here stimulates the cells of the stomach, the G cells specifically, to produce and secrete gastrin. Okay. The second thing, part B here, part B, gastrin does two things. It is going to stimulate parietal cells to secrete pepsinogen. And it's also going to stimulate chief cells to secrete HCl, or hydrochloric acid. So that's what's happening right here in part B. We're stimulating the cells of the stomach to produce these two things, right? The parietal cells produce pepsinogen right here, and the chief cells produce the HCl, the hydrochloric acid. Okay, so part C up next. So let's actually check these off as we go through them. So we've done A, we've done B. So we've done A, we've done B. Next up is C. So C, HCl, of course, is a strong acid, and it'll bring the pH of it'll bring the pH down to really low, about 1 or 2. And what that'll do is that'll do three things. It'll kill any bacteria or foreign particles. Okay. It'll also denature the dietary protein. So the dietary protein that came in here, this little gray blob, that is going to be denatured right here in part C, right? So here we have the H+, the acid, stimulating the denaturing of this protein into a polypeptide shown here. Okay. And the third thing that HCl does, right, that acid does, is it stimulates pepsinogen to auto-cleave itself, to basically cleave itself into pepsin. Okay, so pepsinogen is an inactive precursor to the protein pepsin, which is a protease. Okay. Speaking of which, that ends part C. Now we're on part D. Pepsin, what it's going to do is it's going to, let's actually check off the C over here on the actual diagram as well. So that part there is done. So then part D, the polypeptide, the denatured polypeptide right here, that is going to be cut up into many smaller peptides by pepsin, the active form of pepsinogen. Okay. So pepsin will take that denatured polypeptide and cut it up into little pieces that are going to be small peptides. Okay, so that's part D there. Okay, cool. So then we've got part E. Okay, so now the peptides are going to move into the duodenum of the small intestine and stimulate the duodenum to secrete secretin. into the blood. Secretin. Can I fit that there? Yeah. Okay. And also, it's going to stimulate the duodenum to secrete cholecystokinin, or CCK, into the blood. Okay, so that's right here. The part E, just these small peptides moving out into the small intestine from the stomach. They're going to stimulate the small intestine to secrete those two hormones into the blood. So that's part E. Okay, so now we get into F. Let's check off E over here. Done with E. So F, there's two parts to F. F1 is basically going to be what secretin does. Secretin will stimulate the release of bicarbonate, HCO3-, from the exocrine pancreas. And what that's going to do, that buffer will bring the pH back to around 7 or 8, because it was, that gastric juice that went from over here in the stomach into the small intestine is highly acidic, so bringing that pH back to physiological pH is important. Okay, so that's F1, right? And we got that bicarbonate right here. Okay, so F1, done with that part. Bringing the pH back to 7 or 8. And then F2 is what's going on with the CCK. The CCK, the cholecystokinin, it stimulates the exocrine pancreas to release zymogens, which are inactive precursors of enzymes. So F2 over here, we've got the pancreas dumping the zymogens over here into the small intestine. Okay, so which zymogens are they specifically? One of them is trypsinogen, so it ends in ogen, meaning that it's a zymogen. We've got chymotrypsinogen, again, ends in ogen. We also have got proelastase. Okay, so this one doesn't end in ogen, but it begins with pro. Generally, if it begins with pro, it's a proenzyme. The difference between zymogens and proenzymes, they're pretty much the same thing. They're inactive precursors to active enzymes. And the last one here are, the last two are procarboxypeptidases A and B. Okay, so those are inactive precursors of the carboxypeptidase A and carboxypeptidase B. Okay, all right. And then part G is the zymogens are then cleaved into active proteases. So that's what's going on here. Okay, so these zymogens are all inactive precursors of enzymes that are actually going to act as proteases to cut up these peptides into even smaller peptides. Okay, so the trypsinogen is cleaved by enteropeptidase to become trypsin. And then that trypsin will go through and cleave all of these other zymogens into their active form. So chymotrypsinogen will be cleaved by trypsin to become chymotrypsin, and then proelastase will be cleaved by trypsin to become elastase, and procarboxypeptidases A and B will be turned into carboxypeptidases A and B. Okay, so that's part G there, getting all those zymogens to be active. And then part H, of course, is those proteases are going to cleave the peptides into even smaller peptides. So that's what's happening here. We take these little pieces and cut them up into even smaller pieces here. So now we got really small peptides. Okay. The next step is having the carboxypeptidases removing the carboxy terminal amino acids and aminopeptidases removing the amino terminal amino acids. Okay, so these small peptides basically we're going to remove amino acids off the ends until we have just a collection of amino acids. Okay, so at this point we've taken what we started off with as a large dietary protein that was turned into a polypeptide, and now we've got amino acids from it after cleaving it a bunch of times. So then what happens is so that was step I. So I is done here. And then part J, amino acids are absorbed from the intestinal lumen into the intestinal epithelium. So this portion in here, the actual contents of the tube that is the small intestine, this portion inside is called the lumen. And the intestinal epithelium is the actual cell layers that make it up. So the epithelium is this part here. Epithelium. Okay, so we're going to take those amino acids. They're going to go from that space that is the lumen. They're going to go into the actual cells of the small intestine. And so we get these amino acids. So if we kind of zoom in on that portion there, we've got the amino acids in the lumen. Right out here is the lumen. And they're going to come into the epithelium, right? The cells in here. This is the actual cells of the small intestine. So now we got the amino acids right in there. That's part J. And then parts K and L, those amino acids are going to be taken up from those cells into the blood. So that's what's happening here, right? And then from the blood, they're going to be taken. They're going to be taken in the blood to the liver. Okay. To the liver. So that's parts K and L right there. Parts J, K, and L. And then once the amino acids get to the liver, they can of course be metabolized. Okay. I have a question over here about this situation with these proenzymes and their active forms. Why is it that these proteases are made, stored, and released as inactive precursors? The answer is that so they do not destroy the pancreas. Right? Because if we have the active proteases in the pancreas, they'll just tear apart all the proteins in the pancreas, which of course can be really bad. In fact, that can lead to pancreatitis, which apparently, I've never had it before, but pancreatitis, which is apparently super painful and actually potentially lethal. So you shouldn't fret too much though, because the pancreas has a protein called pancreatic trypsin inhibitor, which basically inhibits trypsin, which is important because trypsin activates a lot of these zymogens into their active forms. So if that can't happen, you won't have a bunch of active proteases in the pancreas, which is good because then, you're preventing havoc from being wreaked on your pancreas, which is pretty awesome. Pancreatic trypsin inhibitor is pretty much the real MVP here. Anyway, if you have any concerns at all about pancreatitis, this is not medical advice in any way, shape, or form. I'm not a medical professional. If you have any questions about that, please go see your doctor. This is all purely approached from an academic perspective. Speaking of which, I hope this video was helpful in getting you to understand how we take a dietary protein and break it down into amino acids that can eventually be taken up into the blood to the liver. Hope that video was helpful. Thanks for watching. If you found that video helpful, don't forget to like, comment, subscribe, and share with friends. Thank you and happy studying.